going to talk about sleep, a behavior that I don't need to introduce. We all engage in daily, and it is required for a bunch of different physiological and cognitive functions. But what I would like to argue today is that its functions are not rigid, but incredibly flexible. And it's this adaptability that can give organisms tremendous flexibility to survive in different environmental conditions. So before going into that, do we even get 7 to 9 hours of recommended sleep every day? So these are the bunch of studies. I don't know how much you can look at it, but essentially these are done across different countries with the help of activity monitors to figure out sleep patterns in different populations. And it was observed that in countries that are typically rank higher on the happiness index, there the population tend to get 7 hours of sleep every night. But if we look at India specifically, that number drops down to 6.4. Another interesting aspect that was discovered with these data sets is that the time to go to bed and wake up strongly correlates with the amount of sleep that you get. So if you go to bed early and wake up early, those are the same countries which were getting 7 hours of sleep. The only anomaly is Russians. They go to bed late, wake up late, but still get a lot of sleep. So other than them, most of the other countries are following these trends. Now, humans are not unique to get sleep. In fact, organisms across the animal kingdom show some sort of sleep-like state, going as low as invertebrates, which can sleep for 2.6 hours in worms, to ants sleeping for 5 to 9 hours, to some species of bats that can sleep for 20 hours in a day. So these are defined on various criteria, but what is clear is a sleep-like state is visible across the kingdom. But still, the behavior remains enigmatic because we still do not completely understand what exactly it is doing. And we have some clues. If we don't sleep, what will happen? So most of the sleep manipulation studies suggest that if we don't sleep properly, then there will be impairment in the bodily functions, including cardiovascular health, immune function, metabolism, and also have effects on brain functions, including learning and memory and social behavior. Even at a cellular level, there are various studies showing some or the other defect of not sleeping properly. So I will be concentrating on learning and memory. We largely try to understand how this link between sleep and learning and memory is established. And the link is in fact reported quite almost 2,000 years back by a Roman educator named Marcus Quintilinus. So he observed that the strength of memory, which seemed weak, was suddenly strengthened the next day on tasks that were not easily recalled on the spot. So this is not directly implicating the role of sleep, but in fact some process that is happening during the night, which might be relevant for this type of learning and memory. But the experimental evidence for the role of sleep in learning and memory was only obtained like 100 years back. In this set of experiments, graduate students were given certain nonsense syllables to remember, and then were either allowed to sleep or kept awake. The group that was allowed to sleep was better at recalling those nonsense syllables, but the group that was kept awake was easily forgetting those syllables. So this gave first evidence that sleep might be playing some sort of role in either the consolidation of the memory or the recall of the memory the next day. So similar studies in other model systems further strengthen this idea that sleep and memory consolidation processes are linked. And we study these processes in the humble fruit fly, a genetically powerful model system, and essentially have contributed immensely to our understanding of various behavioral neurogenetics. So first, I would like to convince you that these tiny flies are capable of learning and sleeping and how actually we go about measuring those parameters. So for learning, these apparatus were developed by Seymour Benzer at Caltech in early 70s. And these are countercurrent devices which take advantage of flies' ability to move towards light. So they are really attracted to light. So they introduce flies here and there's a light at the top. So these flies are going towards the light. At the same time, there's an electric grid that is placed in some of these tubes. And there's a specific odor, a specific smell, which is associated with each, a different smell with each tube. So the experiment is quite simple. You introduce the flies at the bottom, and when they go up, the odor, in this case odor A, will be presented at the same time the electric grid is giving them shocks. So they will form an association between the two. And then there's a second odor where the electric grid is no longer giving them shock. So they will form different memories for different odors. So when they tested their memories, they found that the odor that was associated with electric shock, they avoided that odor significantly compared to the control odors, giving the first evidence that flies can remember this association between an odor and a punishment. Now, how about sleep? So we cannot put electrodes on. We can actually, but recording EEG is not feasible in fly system. So instead of an electrophysiological definition of sleep, we rely on a behavioral definition. So we have defined certain criteria that are essential for sleep, including periods of immobility, a specific posture, an increase in arousal threshold, and a homeostatic regulatory mechanism. I'll come to these in a moment. So the way we do it is we introduce flies in a small tube, and there's an infrared beam running through the center. So whenever fly is moving, these beams will break, and that will be counted as a measure of activity. And if flies are inactive for a period of time, then we take that measure as sleep. So as you can see here, flies typically sleep diurnal. They sleep throughout the night and also obtain some siesta-like sleep during the middle of the day. Now, what about the other criteria? So they do change their posture while they are sleeping. They typically move towards the food and relax. Their arousal thresholds also increase. So here we are using some vibratory devices which will give different strength of stimulus. And when they are sleeping, the strength of that stimulus is higher. So it takes much more intense shaking to wake them up, indicating that they are not awake but in some sleep-like state. And finally, the homeostatic rebound is based on the idea that if you keep them up all night, the tendency to fall asleep would be higher the next day, a behavior that we all experienced at some point. So here in flies also, if we deprive them of sleep with the help of some mechanical shaker, the next day they sleep much longer, which indicate that there's an homeostatic rebound resulting from our manipulation. So like other model systems, sleep is incredibly important for various behaviors in flies. For infection and injury, if you infect flies with a pathogen or you injure them, you see there's an increase in sleep in those flies. And if you don't allow them to sleep, then their ability to recover from the infection is significantly deteriorated. If you don't allow the fly to sleep at all, then they die much faster compared to the controls. In addition, the cognition also changes. Their attention span is lower compared to controls, and their ability to form memories also changes based on how much sleep that they are obtaining. Now coming to this idea about sleep flexibility, all these functions that I mentioned, of course, sleep is important for them, but sleep as a behavior itself is highly influenced by environmental changes. So if you are keeping animals starved, their ability to obtain sleep decreases, presumably in search for food, as shown here. If you keep the animals, in this case flies, isolated, an experience that some of us may have experienced during COVID. Again, the sleep is significantly lower, and temperature changes also changes the amount of sleep and even architecture of sleep. So all these environmental changes are having an influence on sleep, but then the question arises, how would that change the ability of sleep to cater to all those different functions that I talked about? And that's what essentially we try to study in our lab, how wakefulness would affect those sleep-dependent functions, specifically learning and memory in these model systems. So to study that, we use appetitive conditioning. So I talked about electric shock and odor combination, but instead of electric shock, we use reward, like sugar. So these flies are given sugar with an odor, and then given another odor, this time without sugar. Now, to create an artificial situation where flies will be unable to sleep, we keep them starved. As I mentioned, they are unable to get enough sleep when they are starved, or they are kept fed. And then we, next day, 24 hours later, we introduce them back into this similar device, but now give both the odors together. So based on if they remember which odor was associated with sucrose, they will move towards that odor more, and based on that, we calculate a performance index. A higher the performance index, the better their ability to recall whatever they learned. So what we found is in flies that were kept on food, so completely satiated, if we sleep deprive them during after training, their performance indices dropped significantly, indicating that sleep was necessary for them to form whatever memory that they were forming between odor and reward. In contrast, in the case of starved flies, if we do the same thing, if we sleep deprive now the memory is not affected by sleep deprivation. So even though the training is the same, two different types of memories are formed based on whether they are able to sleep or not, based on feeding conditions here, fed or starved. So not only sleep seems to be dispensable for memory consolidation, but it seems that it is acting negatively on memories in starved flies. So what we observed that these flies were more active after memory consolidation, and if we artificially induce sleep with the help of a sleep-inducing drug, in this case carboxydol, these flies' memory performances dropped. And if we sleep deprive them, we rescued those memory performances again. So not only sleep is dispensable, it seems it's acting negatively if these flies are forming what we call sleep-independent memories. So this was what I just showed you. In fed flies, we saw an increase in sleep and seems to be forming memories that are sleep-dependent. In starved flies, we don't see any change in sleep, but we instead see a change in activity, an increase in activity. And here they are forming sleep-independent or activity-dependent memories. So not only flies, these type of behaviors are also observed in the wild in different animals. Killer whale mothers, when they are typically give birth, they move away from their pods. And while making the return journey, which can expand for 3 to 4 months, these mothers as well as newborns show no signs of sleep, which is strange because that's the period where newborn is learning a lot of new techniques. Garden warblers, which are migratory birds, travel across for months, and during that time they also don't show any signs of sleep. And elephants do the same while searching for food across the African plains. They also show no signs of sleep. Of course, in humans also we have chronic sleep deprivation, insomnia problems, which are exaggerated by our current lifestyles, but a direct effect on physiological and cognitive functions is hard to determine. And I would like to argue that in these cases, a sleep-independent mechanism might be playing a role. So before going forward, I just wanted to talk a little bit about how memories are formed in the fly brain. So this is how a fly brain looks like. When an odor is provided, these antennal lobes, these blue colored structure, first process that information and then transfer it to these higher brain centers called mushroom bodies because they look like mushrooms through projection neurons. So the way memory is established is these are different dopaminergic neurons, PPL1 and PAM. And what they do is they modulate the connections within the mushroom body neurons such that when the odor is presented again, the approach or avoidance pathways are differentially regulated. So that if a reward was presented with an odor, a fly will approach that odor, and if punishment or a shock was associated, then it will move away from it. So what we found was for our sleep-dependent and independent memories, the underlying neural circuitry is completely different. So here, I would not go into the details of this, but what you can appreciate is different set of neurons are required for memories when they are forming sleep-dependent or sleep-independent. Even though, again, I like to emphasize the training is the same. They were both given odor and sucrose, but based on whether food was available, they're forming different types of memories. So here I would like to switch gears and talk a little bit about what we are currently doing. And I briefly talked about temperature, how temperature changes the sleep patterns in different model organisms, and that's also true in flies. Here I'm showing you rodent data, which are nocturnal animals. And what you can see here is during nighttime, there is a decrease in their body temperatures compared to their active periods. And in fact, if the transition from wakefulness to sleep is preceded by a decrease in their body temperatures. And if there is a decoupling between body temperature and sleep-like state, then insomnia-like behavior is observed in these animals. Now, in humans, similar studies through various surveys have concluded that lower temperatures don't have that much of an impact on sleep states, but higher temperatures can result in a significant decline. And that begs the question, what would happen if there is a considerable increase in external weather or even acute temperature increase? An experience that some of us may have experienced, but with increasing global temperatures would become a much more serious issue. So in these cases, the expectation would be some sort of stress response will be activated. In fact, it has been shown that the cortisol levels go up when humans are exposed to a high temperature, even for short periods of time. And we would like to study what would that do to our ability to form memories. Is the function of sleep remaining the same or changing based on these high temperatures? So this was first looked at in C. elegans worms. And what it was observed that if you give them a heat shock, they show an increase in sleep. And if you don't allow them to sleep properly, then their survival is down. So something similar to what we observe with immune stressors, immune challenges. There also, sleep seems to be playing a significant role in recovery, in survival of animals. Now, we have done similar experiments in flies, and we also observe that after a heat shock, there is an increase in their sleep. So first we wanted to look at whether this is also true in starved flies, the ones that are forming sleep-independent memories. And similar to fed flies that show an increase in sleep, our starved flies also showed an increase in sleep after a heat shock. So here they are kept on at 38 degrees Celsius for an hour for heat shock conditions. So next we looked at what would happen to memory consolidation. So flies were trained and then exposed to a heat stress, and then looked at whether that has any effect on their learning and memory. So again, performance indices suggest that in both starved condition and fed condition, just giving them a heat shock resulted in a significant decline in memory consolidation. So in both conditions, even though the sleep is going up, memory consolidation seems to be impacted. Then we sleep deprive and see what happened. And in starved case, we were able to rescue that sleep, the memory decline. But in fed case, we were unable to do that. So it seems the heat stress is differentially affecting sleep-dependent and sleep-independent memories. And our current data suggests that dopamine might have a role to play in how these different mechanisms are activated. So I'll quickly talk about what we found with dopamine. So here we are using a genetic construct to, we have introduced a genetic construct in specific dopaminergic neurons in the fly brain. And what this construct will do is whenever a neuron is active, this will generate a luminescence-based signal which can be read by a plate reader. So what we found is after heat shock, these dopaminergic neurons were more active compared to the control condition. And then to further look at their behavior, we used optogenetics to artificially activate these neurons after training. So here we are not giving them any electric shock, heat stress, but in fact just activating them with the help of optogenetics. And what we found is the performance index is declining, indicating that this neuron, this particular dopaminergic neuron, might be the one which is getting recruited by the heat stress to change this memory pathway. I will not go into the details of these other aspects that we discovered, but what our current data is currently showing is the way heat stress is recruiting dopaminergic system to then increase the activity of a particular receptor, which is also a receptor for a steroid hormone called ecdysone, something similar to cortisol in mammals. So we are trying to figure out how this connection might be functioning, but at this stage, what we can say is the differential effect of heat stress on sleep-dependent functions versus sleep-independent functions might be down to the differential energy requirements for these two pathways. So what do I mean by that is whenever there is a heat stress or a stress of any sort for that matter, to increase recovery from that stress, a lot of energy is required. And to facilitate that system, what our data is suggesting, the system is taking that energy away from processes like memory consolidation. So the decline in memory performances or the impairment of memory consolidation is essentially to facilitate the recovery from the process. And if we don't allow that recovery to happen, if we artificially make them still learn in those heat stress conditions, then their overall survival is getting affected. So this mechanism is allowing to keep this balance to allow an organism to survive, and sleep seems to be playing a major role in this toggle from memory consolidation or survival from a heat stress like stressor. So this is my lab at TIFR Mumbai. Most of the work that I showed you today is done by a graduate student named Kamakshi, and these are the funding sources. And thank you so much for your time.
Thank you for the great talk. It was really very clear and I really enjoyed a lot. And when you talked about heat stress, I was wondering, and since you were also talking about the environmental effects, I was wondering, like the example in like Scandinavian countries, when in the summer it's always light when they're going to sleep, and in winter, so still if you sleep for 7 hours, and by the way, I have to say that is the 7 the new 8, because we have been hearing 8 hours through since our child.
7 to 9. Yes.
Okay, but anyway, so is the sleep quality affected depending upon the external like sun, because temperature is maintained because of air condition. It's just the environmental factor which is different.
So the studies that I showed you, those were Hungarian surveys which were done over 25 years. So yes, modern technology now allows us to maintain our internal room temperatures to an extent which can help us to fall asleep better. But those surveys were started when those technologies were not available. So there we can see that if you decrease the temperature, even take it to negative, it doesn't affect sleep that much. But even a 10 degrees Celsius increase in temperature from 15 to 25 seems to decrease it. And we are talking about temperatures above 40 degrees Celsius.
Sunlight doesn't have any correlation with the quality of sleep then?
I mean, sunlight is essentially giving a circadian signal for them to maintain when to sleep or when to keep
That's
active.
true, but if you take example of Norway, for example, you maybe get sunlight for 2-3 hours, and then in winters, and then in summer you don't get dark when you go to sleep. So that's what I meant actually.
Okay, I mean, it's so whatever I've shown you today, it's largely done with 12-hour cycles. But other researchers have delved into that question to decrease the cycle lengths and see what happens. It drastically changes their behavior, but it doesn't really increase the quantity or quality of sleep. So they become more active, but they then remain active even during the dark phase. So there is a change, but it's not like they are sleeping more just because of that.
I have a question. Is there any difference in the nature or pattern between sleep-dependent memory versus sleep-independent memory?
So with sleep
in terms of long or short-term memory or things like that?
So all these experiments that I showed you are for long-term memories. The sleep pattern, so quality of sleep is much higher for sleep-dependent memories. If you decrease only the quality of sleep, which essentially means fragmenting their sleep, then the memory is affected. For sleep-independent memories, they're completely dispensable. Even if they are not sleeping at all, they will still form that memory.
Okay, we'll move the rest of the questions to the end of the session after Arun's talk. Thank you very much, Nitin.